Hi, this is Vidhi Vaidya and our today's topic about transcription. Now before we study about transcription, we should first have to study what is central dogma of life. Now in central dogma, if a product is protein and the information we are getting from is the DNA. Now to transfer the information from DNA to protein, we have to take two steps which are transcription and translation. These are two processes in which we can convert the information from DNA to protein. In this video, we are going to learn about transcription. Now, what is transcription? It is a process in which the information of the DNA is transferred to mRNA, or you can say the messenger RNA. Why we call this RNA as a messenger RNA? Because it works as a messenger to transfer the information of DNA to the further modification and protein synthesis. Transcription process is in three phases: initiation, elongation and termination. We are going to study each of that. There are terms in initiation I am going to tell you. Now, these are the things which you have to first consider before going to start the initiation process, which are closed complex, open complex, enzyme. The enzyme which we use in this process is RNA polymerase, and this is the initiation site, or you can say the site from which we are going to start the process of transcription. Like if say this is a DNA template, and here is the +1 site from which we are going to add our nucleotides of mRNA. So it is called a +1 site or the initiation site. Now the other sequence we are going to consider is promoter sequence, which is this site of the initiation site, -35 and -10. On this particular site, the enzyme RNA polymerase going to bind. Now, as we know that DNA is a double helix structure and we want only one strand to be transcribed. So what we can do in that case, suppose these are the two strands, but we want only this strand to be transcribed. So there will be one template strand and one non-coding strand. Now, template strand is the strand from which the information is transcribed to the RNA. Now, in the initiation process, as I told you earlier, that there are promoter sequence present on the DNA itself. So suppose that these are the promoter sequence -35 and -10. The enzyme we call the RNA polymerase comes and first binds to this sequence. When it binds to this sequence, the complex formed is we call the closed complex. Now, as the RNA polymerase has bind it, it will start separating these two strands by giving the structures like this. This kind of formation we called as the open complex. Now, this will be the template strand and this is ready to the coding. Now, in elongation, we have right now template strand, the open complex. Here are the promoter sequence. And let's just suppose that this is the initiation site. Now what the RNA polymerase does is that it opens up this initiation site and forms the open complex and then brings the nucleotide here from the first site, or you can say the initiation site, and start producing the mRNA. One thing you have to remember that there will be a protein called protein bridge, which only allows one template base. Let's suppose this is the base, or you can say this is the base. It only allows this base to be exposed, and then RNA polymerase do not confuse between all the bases and hence bring the complementary base for this base. Now, as you know that DNA has bases of A, T, G, C, but if there are A here, there will not be T here because this is RNA and hence it is U instead of T. Now, termination or the stopping of the process are divided into two processes. RHO dependent and RHO non-dependent. RHO is a protein which is responsible for the termination. Now, in RHO non-dependent, what happens is when we are near to the termination sequence present here, there will be GC sequence present on the template and hence the GC sequence present on the mRNA itself. After that, there are UUU present in the end of the mRNA because of the poly U present at the DNA template. Now, at one stage when this comes out, what happens is G and C are both complementary to each other and hence they form bond between them. Suppose this is it will form three hydrogen bonds between them, and there is a formation of hairpin like structure. Now, as you know, the bond between U and A is of 2 hydrogen bonds and this is of 3 hydrogen bonds. The stronger is this. Now, because it has more power, it grabs out this mRNA template and separates these 2 bonds, and termination happens and RNA comes out. Now, as I told you that RHO is a protein which is responsible for the termination. In RHO dependent, there will be a RHO protein. This is the open complex. There will be the mRNA. Now, at one stage, there are so many C's present on the mRNA sequence, which actually attracts the RHO protein, and then this RHO protein actually binds to this poly C site. Now, this RHO protein is actually a rotatory protein which rotates towards the end of the mRNA in a rotatory form and separates the bond between the DNA template and the mRNA. Hence, it grabs out the whole mRNA. Now, after termination, the mRNA which we had collected will go with the information of DNA to process called translation, in which there will be specific proteins or you can say the amino acid produced on the basis of this information. So this is it for transcription. If you have any doubts or if you want me to learn each and every initiation, elongation and termination in details, then please comment down in the comment box. And thank you for watching the videos. Please like, share and subscribe to the Biophilic channel if you like this video. And please do not forget to subscribe to the channel so that you can notified whenever I post any bio tutorials videos. Thank you.